presentation was so both valuable and professional, and the quality of her final deliverable was so good that I didn't think that, you know, like giving the good grade and stuff would be sufficient. So I sponsored, I can't remember how much, but a scholarship to send her to AGU in Washington, D.C., just because it was so good. I wanted Hadley to have the opportunity after showing that effort in the summer to see science at that top level and not have to pay for it, right? A huge registration fee, huge barriers of entry. But I haven't done that before. Performance in a class so good where I'm like, okay, I want you to just go learn more. So I think that, you know, everybody in the group would agree that, you know, Hadley set an example and set a tone. And I also want to throw in that she is from the Jersey Shore. And there's family of hers that is probably within 2 or 3 miles from where my dad lives. So the Jersey Shore is an interesting place and very well represented here this year and every year. And our next presenter has done a lot of very interesting and cool things, but this particular project relates to a super interesting transboundary issue related to water, identifying a spectral signature of untreated wastewater from the Tijuana River. Hadley, the floor is yours.
Thank you, Corey. I'm actually not from the Jersey Shore, but I do have family there. So I've spent a lot of time there. Corey's also the one who initially gave me the idea to pursue a topic related to this issue. So thank you, Corey, for inspiring this. I am Hadley Menk. I'm going to be talking about my project on identifying a spectral signature of untreated wastewater from the Tijuana River. So I'll just go over throughout this presentation some of the context and background for why I chose this topic, some of my methodology, some initial results, and some discussion of what these results might mean, and then I'm going to talk about some applications and potential next steps. So just to provide some context about this issue, untreated wastewater pollution entering the environment in the San Diego-Tijuana border region has been an issue for the past several decades, but it's become particularly acute in the last 5 years or so due to some really major infrastructure failures, particularly the failure of a treatment plant on the Mexican side of the border 5 years ago that has been since closed for repair up until a couple of weeks ago. And that resulted in untreated wastewater, instead of being treated by the plant, being discharged directly into the Pacific at Punta Bandera. There's also been a large-scale discharge of untreated wastewater directly into the Tijuana River due to insufficient treatment infrastructure, and that also finds its way into the Pacific Ocean. So there's a huge combination of factors for why this is happening: population growth in Tijuana, economic growth, and just insufficient infrastructure. So untreated wastewater, it's a global problem. It's not just affecting these communities, but it also has been associated with pathogens that are harmful to human health, including hepatitis A, COVID-19 virus, norovirus, and some bacteria, including E. coli. There's also been really prolonged beach closures in Imperial Beach, California, and that has led to some pretty damaging impacts on the local economy there. And there's been human illnesses that have been attributed to this release of untreated wastewater. So I just pulled out some headlines from national news sources that are fairly recent because this has been getting some national attention. So this is a graphic from the U.S. Environmental Protection Agency, and what I just want to point out here is the 2 point sources of this untreated wastewater. So the Tijuana River, which is a transboundary river that flows across the border and then goes through the Tijuana estuary, which is a protected estuarine reserve, and then discharges into the Pacific Ocean just south of Imperial Beach. And then the other point source is down here at San Antonio de los Buenos, and this is where it discharges directly into the Pacific. And importantly, during the summer, it's very common to have a northward current in the Pacific, which is transporting this pollution up the coast and impacting communities in coastal areas in Imperial Beach and San Diego County. So as you can probably tell, this is a really complex problem. It's not just a scientific problem, it's also a political one and an infrastructure problem. And so just some recent, there is a lot of impact, incentive to try to fix this problem. What I should mention in this figure is that there actually is an international treatment plant, and that is run by the International Boundary Waters Commission, and they are planning to expand and double the capacity of that treatment plant in the next 5 years. The Punta Bandera treatment plant also, as of a few weeks ago, has started treating water again, and that hopefully should help alleviate some of that discharge at Punta Bandera. However, the Tijuana River is probably still going to continue to have this problem, particularly as climate change worsens extreme precipitation events in the area. That has the potential to overload the existing infrastructure and cause untreated sewage to be released into the environment. And this happened a couple of years ago during Tropical Storm Hillary. There was really a lot of rain in a short amount of time, and that causes the infrastructure that is already overtaxed to really just not be able to handle the volume of stormwater and wastewater. It discharges into the environment, and flooding also spread the pollution beyond where it normally lies. And so these sorts of events, these extreme precipitation events, are expected to become more frequent in San Diego. And this is also going to be a problem not just in this region, but globally as climate change accelerates. So my project focuses on remote sensing and potential applications of remote sensing for detecting wastewater pollution in coastal environments. So remote sensing is essentially using satellite data to try to track wastewater pollution. There's been a couple of studies in Southern California that have tried to tackle this. Notably, the study from 2020 by Ayad et al. Was able to distinguish wastewater plumes from stormwater plumes using satellite data off the coast of San Diego. And then what's really challenging about this problem is there's a lot of complexities in the water, and so it can be really hard from satellite data to distinguish those two things. So the way that satellites essentially work is by reading the reflectance of light off the ground or off the water. And so if you can kind of discern those patterns in where light is absorbed and where light is reflected, you can try to work out what you're looking at with the satellite. So a study last year lead authored by Eva Scrivener at SDSU tried to tackle this problem, and they basically collected untreated wastewater samples from the treatment plant, diluted them with seawater, and ran them through a spectroradiometer, which basically just measures the reflectance of light in the same way that a satellite would. And what they found is that in the wastewater samples, there was a spectral feature centered at 620 nanometers. And these dips in the graph mean that something is absorbing light at that wavelength. And so what they found was this dip, this spectral feature at 620 in wastewater samples that potentially could be used to try to identify wastewater using satellites. So my work really builds off of this study and tries to extend it using environmental samples. So the goals of this project were to build on that existing work. As I mentioned, their samples were from a treatment plant. The samples that I'm using are from the environment. Also helping to validate some existing satellite plume tracking work going on at SDSU, understand some of the spatial patterns in my water quality data, and explore some future remote sensing applications. So before I get into my methods, I just want to acknowledge that this was very much a group effort. I could not have done it without any of the people on the slide, but particularly my committee chair, Dr. Dan Souza at SDSU. He has a grant from NASA to do this work and has been so supportive and encouraging throughout the whole process. Dr. Sarah Giddings is a coastal oceanographer here at Scripps, and she is involved with the Pathogen Forecast Modeling Project, which has built a forecast model to help inform residents about pollution risk. And I also just want to shout out Alex Grant and Sneha Karat at SDSU, who ran a lot of the water quality analysis for this work, and Dr. Jeff Bowman and Shelby Marhofer-Jess at the Bowman Lab here at Scripps. They provided me with samples that I used in this project, and also lab space and just a lot of great resources and support. So getting into what I actually did, this is a map of where the sampling locations that I had were. I should probably say that the title of this project is kind of misleading because I also was able to use samples not just from the river, but also the ocean the estuary, and the treatment plant. So the Bowman Lab was able to provide me with samples from offshore, off the coast of San Diego and Imperial Beach from boat-based sampling, and also beach samples at Borderfield State Park. And then I have a couple of river and estuary samples, most of which were provided by SDSU, San Diego State University, and their ongoing monitoring program. And then I also collected some of my own samples at Imperial Beach Pier and at the river. Once the samples were collected, I then brought them to the lab and I analyzed them with a spectrophotometer. So I used this NICS Spectro 2, which is this little portable low-cost sensor, and basically just put some water in this cuvette and placed the NICS directly on the glass cuvette and scanned it. And it's measuring the reflectance and absorption of light, like I mentioned, in the visible spectrum, which is between 400 and 700 nanometers. And then the samples were analyzed in the SDSU lab for some key water quality variables. I chose to just focus on 2 for this work, both because I had this data for every sample and also because they're important for remote sensing or could be potentially important. So tryptophan is a wastewater indicator, and it has been shown to be highly correlated with E. coli, which is a fecal indicator bacteria, and CDOM, which is basically just like colored stuff in the water. That has already been used in remote sensing applications and has been correlated with E. coli concentrations, specifically in the Tijuana River. So some initial results. From just the water quality analysis, I in total analyzed 147 samples across 11 dates in the spring of this year. And what I just want to point out here is this is a logarithmic scale and just some average min and max figures for these 2 water quality parameters. It's kind of what we might expect in that the ocean samples had the lowest mean and max levels of both of these indicator quality parameters, and then the estuary is the second highest, the river, and then the treatment plant, which, as you'll see, this is untreated wastewater before it's been treated. So it is concerningly similar to the river samples. The other thing that's important to point out here is that the ocean samples in particular have the potential to be influenced both by the river inputs and by the Puta Bandera inputs. So it's important to note that there's 2 potential sources there. But as the water moves through the system, it's being diluted by clean water, which is why we might expect to see this decline. So the pathogen forecast model is basically a physical model that is predicting the movement of this water. So this is provided by my advisor, Sarah Giddings, and this third pane is the Tijuana wastewater fraction. So this water quality data has the potential to help potentially validate this model. It also kind of is in line with what the data shows, in that as the water moves further offshore, it's being diluted and the levels are dropping. So the next thing I did was I plotted some of my spectra. This is what it looks like when you plot a spectral graph. And I just pulled out this one sample, which is an unfiltered Tijuana River sample, to just show some of the dips and the peaks. It's important to mention that, like with water, it's very difficult to get spectral data. A lot of the changes are super subtle. But I just wanted to show this one because it had some features that were sort of characteristic of a lot of the wastewater samples, or the ones that had high CDOM and tryptophan. So we have this sort of reflectance peak at 550, and then a very slight dip around 620, and then a very slight increase or peak at 650 where something's reflecting. But it's super subtle and it's very hard to like pick out with the eye, which is why I tried to do some regression analysis and statistical analysis. This is just kind of a very preliminary step to try to pull out some initial patterns, and then hopefully I can continue to build on this work and dig more into the data. But I used a machine learning model, a couple of them actually, and I'll focus on the partial least squared regression, which is very commonly used in spectral data. So the first thing I did was I trained the model to get the right number of components, and then what I tried to pull out was basically the model using the data is then predicting CDOM and tryptophan concentrations, and then giving me back an input of how much the different spectral features or wavelengths might be influencing that, or not influencing, but like correlated, I guess is the right word. So I just tried to pull out the top 4 from each. This was from all the samples that I analyzed together, and then I just wanted to point out that 610 is showing up in both of these, as is 520. And then I also separated the filtered and unfiltered samples. The unfiltered samples I think could be more interesting potentially because this 620 wavelength is showing up again, and that is what the previous study used, was unfiltered samples. So just to discuss what some of these initial findings might mean, I want to caveat by saying that a lot of the models, some of them were not particularly well fitted to the data. They need some more tweaking, but this can hopefully help bring out some initial patterns. So the 620 correlations, that correlated to high CDOM and tryptophan concentrations in the unfiltered samples only. So it did not appear in the filtered samples, which could mean that something there in the filtering process is removing whatever is absorbing light at that wavelength to the extent that it's not showing up in the filtered samples. But the tryptophan ones in particular had a very low R-squared value, which means that the model is not super well fitted. So that needs some working out. And then 610 appeared in most of the models, which was also interesting. For CDOM in the all samples analysis, 440 and 670 also appeared, and as in the top 4, and those are supported in literature, and they've been used as spectral signatures of CDOM to try to derive CDOM concentrations from satellite imagery. And the other important thing to note here is that these are environmental samples, and it's super hard to pick out what is actually causing the light to be reflected or absorbed at each wavelength. There's a lot of potential inputs, not just wastewater, also urban runoff, natural inputs like sediment that could also be impacting here. So that's just important to note and would need a lot more work to sort of unpack. So some key findings and future applications. The spectral feature at 610 correlated with CDOM and tryptophan in the models. The 620 feature appeared in the unfiltered samples. The 610 feature is not clearly explained by prior literature, but as I mentioned, some of the models were not super well fitted to the data and were kind of telling me different things. So I want to further unpack that potentially. But if we're able to potentially find a spectral signature of wastewater, that has a ton of applications, both for satellites that are currently orbiting, including PACE, which was launched last year, and is using the same wavelengths of light in the visible spectrum, and also hyperspectral satellites that are currently in development. This could be a potentially really useful application of those. And remote sensing can be a really valuable addition to monitoring programs in coastal communities. It is low cost, it's low, relatively easy to use. It can help fill in some of the spatial gaps in existing sampling programs, which are very expensive and time consuming. It can also fill in temporal gaps. We're not able to be out in the field sampling all the time. So if we can use remote sensing, it has the potential to be a really powerful tool for coastal communities that are being impacted by this problem. So that is it. I just want to thank, again, my committee, who was absolutely incredible. Acknowledge that this work was funded by a couple of NASA grants that Dan Souza has in his research group. And just thank my friends and my family and for all of your support. I could not have done it without all of you. So thank you.
Really fantastic. The question has to do with the estuary and whether the spectral monitoring could detect whether plant evapotranspiration is absorbing any of the sewage into the plant material. And if so, how does that change?
That is a great question. I know that there are satellites that actually can do that and that do measure evapotranspiration. My initial project idea was actually to use an instrument that can measure that, and it mostly measures heat on Earth. So that would actually be a really interesting question. I don't know if there's actual satellite applications yet of plant evapotranspiration in terms of pollution specifically, but that is a great question. I would love to look at that further.
I was just curious a little more about the significance of why this pollution in this area matters, and what are some of the concerns that you took away from your findings as far as that goes?
Yeah, that's a great question. Thank you, Cassie. So the main significance is just the public health effect. So this has been making people sick in these communities, not just beachgoers and people that are swimming in the water, but people that are living there. There's a paper that was published literally like 2 weeks ago about potential aerosol interactions with the pollution and how people breathing just in the air in this area could be making them sick. So the public health impacts are super important. It also, as I mentioned, has a severe economic impact on the tourism industry here. It has obviously pretty bad ecosystem effects for marine life in particular as well. So there's a huge host of problems, and this is not just an issue here, it's an issue in a lot of places, particularly developing countries, but also just all over the US as our infrastructure ages. Thank you.
Excellent job, Hadley. As those of us who have been around San Diego for a while, this is Tijuana.